Okay, so welcome everyone. My name is Rea Globus. I'm the Business Development Manager at YDL Ventures. I want to thank you all for joining us. Just a quick introduction. This meetup is a part of YDL's drug development series of meetups that takes place every month, covering different areas of the drug development process. So today we're going to discuss drug discovery trends and new insights from phenotypic approaches. We have two speakers. We have Alistair King and Cheryl Denker, and I'll let them say a few words for the introduction.
Yeah, hi, thanks, Rea. So I'm Alistair King. I'm the head of biology at Eurofins PanLabs, which is part of the global Eurofins Discovery Organization. My role really is scientific oversight over all of the services that we offer here, and it's a pleasure to have the privilege to be able to speak to you all today. And pass over to my colleague Cheryl.
Thanks, Alistair. Hi, everyone. I'm Cheryl Denker. I'm a cell biologist by training, and I'm working with the Phenotypic team, sort of at the nexus of the product marketing, Marcom, scientific, and business development teams. So thank you for having us today.
Thank you both. So just before we begin, I want to say that the chat is open, and feel free to send us questions, and we'll try to address everything by the end. And that's it. You may begin.
Well, thank you very much, Ray. Like I said, it's a pleasure to be able to speak to you all today. What we're going to discuss will be latest drug discovery trends, and in particular, how that ties in with phenotypic approaches to drug discovery. So with that in mind, this is the agenda for what we'll cover today. We'll talk initially a little bit about some of the current trends in general drug discovery. And in particular, obviously, because we've all been impacted by this over the last couple of years or so, the impact of COVID-19 on drug discovery, because it definitely has had an impact, and we feel will have an ongoing impact in terms of new agents and how things are developed. We'll then talk about some of the advances that have been made in phenotypic drug discovery, in particular approaches and technologies that are used, and how and where those approaches have been used. And we'll also talk in terms of a case study. So my colleague Cheryl will present an overview of a case study there around fibrosis, and showing the power of phenotypic approaches and how that can really provide benefit in a drug discovery environment. And then at the end, we'll tie up with a summary of what we've discussed. So with that, then, we'll start with an overview of current drug discovery trends, and we'll discuss a little bit about how COVID-19 has impacted the general drug discovery world. So as you can see from this slide, in the last 20 or so years, there's been a steady increase in a number of things in drug discovery. Throughout the pandemic, drug discovery and approvals have been relatively steady, but there has been an increase, you'll see here in the red bars, in biologics drug discovery, really for the last 10 or so years. This has been driven by a number of things, in particular in, for example, the oncology field, the approval of agents that target the immune checkpoints. And that has created a lot of interest around development of new biologic therapeutics there, but also advances in the inflammation world, as well as other therapeutic areas, have helped drive that collective biologics drug discovery. In addition to that, we'll talk a little bit about this a little bit later, are some of the newer modalities that are tying in with approvals here. Now, you can see overall the number of biologics as a percentage has increased. It's a little bit like the stock market here. It goes up and down, but the general trend is on the increase. And then you can see that as the number of biologics here as a component of the overall drug discovery approval. But you can see otherwise it's been fairly consistent. And you can see in particular here throughout the pandemic, overall approvals for new molecular entities has been fairly consistent, which is good to see, obviously with some of the trials that we've been going through with the COVID-19 pandemic. Now, I mentioned about biologics drug discovery. Obviously, this is an area that is very much on the increase. And we can see that really here with a snapshot of 2021 and some of the biologics that were approved in that particular year. So collectively, biologics made up a little bit less than 40% of the overall approvals by the FDA in 2021. And of that, antibodies represented 20%. So that's sort of broken down in the pie charts at the right-hand side here. So you can see that antibodies, monoclonal antibodies in particular here, remain very strong as a major component of the biologics approvals. Tied in with that, of course, are other antibody therapeutics, for example, antibody drug conjugates here, bispecific agents, but also other biologic agents outside of the antibodies, for example, enzyme therapeutics and other formats. Now, not to forget, it's grouped here within the small molecule area are other variants effectively on small molecules, in particular peptides, that are sort of in that crossover realm between small molecules and biologics. In some cases, they are sometimes considered biologics. In this particular case, we're considering peptides up to 40 amino acids. And then last but by no means least, the oligonucleotide therapeutic modalities, so antisense RNAs or siRNA approaches. And this is an area that is very much on the increase as part of an emerging trend where one is seeing new therapeutic agents that are coming onto the scene. And again, we'll talk a little bit more about those and how they tie in with phenotypic drug discovery and the need for phenotypic drug discovery in collectively a lot of biologic approaches, especially some of the emerging technologies. And just a couple of examples here of some of the most recent approvals. Now, when one looks at the therapeutic areas and breaks this down by that, one can see that there have been pretty steady approvals, as we saw on the last slide, certainly as far as these 2021 data go. And these are data looking at approvals in 2021 over the prior 5-year average. So you can see in certain cases, oncology is obviously still leading in terms of the area of major focus for drug discovery. Maybe slightly more approvals in 2021, but essentially a very similar approval rate. Same kind of thing in certain other areas. Two areas do stand out, albeit the numbers are relatively smaller, but they are notable, and that is cardiovascular. Experienced more approvals than it had over the previous 5-year average, as did psychiatry. So showing tremendous strides in both of these therapeutic areas, again, in the face of relatively consistent, or in a couple of cases, infectious diseases and neurology showing a slight decrease there. The irony here, obviously, with COVID-19 being that infectious diseases did see a decrease. One wonders, one speculates going forward as to the approvals that will come about as a result of COVID-19. Perhaps in the medium to longer term in the virology area, as we get to consider the importance of antiviral therapeutics, and also in the inflammation area. As we've seen over the last couple of years, there's been quite a bit of interest in looking at inflammation and how those therapeutics can be used to treat some of the effects of COVID-19, which of course results from SARS-CoV-2 infection. Just to look here in terms of biotherapeutics, so phenotypic needs are important in this because as we go forward, and we've come over the last several years, and these trends, I think, will continue. We've seen the emergence of a few new therapeutic modalities, so things like gene editing and gene therapy approaches. We see things like cell therapy approaches, and then closely tied in with that are things like CAR-T and NK cell approaches, as well as the RNA approaches and other approaches that are being examined. But you can see quite clearly here from the CAR-T and NK cell, the cell therapy and the gene editing therapy approaches, that these have been very much on the increase since about 2016, and they are set to increase even further. Now, because of the complexity of these are obviously very different from conventional small molecules or even antibody therapeutics that in of themselves are relatively selective. These represent quite complex therapeutics and thus involve potentially complex biology. And so, as we'll see as we go through the talk, this is really where the phenotypic drug discovery approaches that we can employ really come into power in evaluating those aspects of complex biology and, in some cases, polypharmacologies. So in terms of where we think the drug discovery area is going, there are obviously advances in a variety of different therapeutic strategies. We talked about some of those on the previous slide. But even in small molecule or biologic areas, as I'd mentioned on one of the previous slides, antibody drug conjugates, we are seeing more approvals in that area. Obviously combining the benefits of small molecules and antibody therapeutics to deliver what might otherwise be more toxic agents in a very targeted manner using the selectivity of antibody therapeutics. Obviously, the acceleration here, by evaluating human in vitro models is important in understanding how these agents work. And as I'm mentioning here on the middle paragraph of this is especially true when comparing approaches with potential severe adverse outcomes. So as we get into some of these newer therapeutic areas, like I said, they're a lot more complex than traditional therapeutic approaches. We have to consider wider aspects of the biology. And in the case even of things like antibody drug conjugates here, where, as I say, you're employing the selectivity of a monoclonal antibody to deliver what is in most cases a very potent cytotoxic agent, one has to consider the phenotypic approaches there and how that can impact potentially severe outcomes to make safer drugs. And then obviously gene-based therapeutics here also become more complex because as one knocks out a gene, there are wider and broader consequences there than perhaps use of much more targeted agents. Now, an important part of using phenotypic drug discovery approaches, and especially in these complex biological scenarios, is that they can very much enable quick win or fast fail outcomes. So what we'll see as we go through the presentation is the employment of phenotypic drug discovery approaches earlier in the drug discovery continuum allows a number of different benefits. That includes evaluation of novel biologies or some of the complex biologies that may not be immediately obvious from more targeted traditional in vitro drug discovery approaches, to do that earlier. So then one can potentially identify things like safety or toxicology issues earlier and do something about that before one goes into clinical testing. Obviously, if one finds that out right upon going into clinical testing, that it can either hold up that testing and potentially doom a candidate, or even worse, finding out the issue during clinical testing, which of course then will severely impede the progress of a particular program and possibly even halt that trial. So just an overview here as to why and where one should use phenotypic drug discovery approaches. Obviously, we're all familiar with the process of going through screening and then going through the drug discovery pathway to look at things like efficacy, safety, and in many cases, especially as one gets towards candidate selection, the superiority of a drug against current standards of care. But the goal isn't really to achieve any one of these. Obviously, it's important to achieve all of these because they're all very important for getting approval to go into clinical trials. But ultimately, the goal of drug discovery should be clinical adoption, because for a drug to be superior to that which is on the market or even a new medication, it has to be approved and it has to be used. So a drug really should be the best it can be. And one of the ways of evaluating this is evaluating that more complex biology, which is affordable by using phenotypic drug discovery approaches. So now we're going to take a quick look at some of the new advances in phenotypic drug discovery approaches and why they are important in drug discovery. So going now into a little bit more detail. So if we look at traditional drug discovery, target-based drug discovery, typically it follows this kind of path where you have compounds, or in the case of even biologic agents, looking at panning phage display libraries and things like this, to use selective targeted assays. These may be recombinant proteins, or they may even be cellular assays that are engineered that have a particular target in them, but one is looking solely at the activity at that target. So a fairly logical and conventional approach to identify hits that modulate the target of interest. And this, of course, is once a target has been validated for its association with a particular disease. And then showing in things like a cellular approach that one has target engagement. So maybe from a biochemical assay here, showing that your hits that come out of that do in fact modulate the target in the slightly more complex environment of a cellular assay system, where perhaps the conformation may be slightly different, or the context of the target may be slightly different, for example, within a complex of other proteins in the cell. And then showing target selectivity, so typically doing counter screens. A good example in the oncology field might be showing target engagement for modulation of a target in a particular tumor cell, but then showing selectivity in a broader sense there. Other examples of this may be looking at, for example, in kinases, where you are impacting the activity at one kinase, but not other kinases, and in particular, perhaps closely related kinases that you do not want to impact or modulate with your agent. And then ultimately looking at the biological impact. So that may take the form of what we've typically called functional assays, but otherwise phenotypic assays, to see that having developed an agent that is targeted and you've shown cellular activity, does it have the biological consequence that you wish? And especially in terms of the disease state, does it reverse the disease state? So, for example, does it inhibit human tumor cell growth or viability? Does it modulate cytokine secretion in an inflammatory system? So this is the traditional target-based approach. So really, how does a phenotypic drug discovery approach differ from that? Well, one still starts with an array of compounds or biologics in something like a display library. But one invokes biological systems much earlier in the pathway. So obviously these bring in a complexity with them, but from that you are able to evaluate biological impact sooner. So collectively these two steps might include some of the steps that you are looking at later here. Now, in these particular cases, sometimes there has been concern over conducting SAR development studies using biological pathways, because obviously they are more complex. And there is always a concern that any changes you see might not necessarily be related solely to activity at the target, but in some cases related to polypharmacology. However, it has been shown in numerous occasions that one can develop SAR around the biological impact itself. And after all, since the biological impact is ultimately what one wants to see in a therapeutic setting, this is important to evaluate this earlier. Not only can it show activity in the context of a disease setting, but also potentially identify safety issues earlier that can then obviously be addressed before it is the program is in a much later stage. One can then go back and look at the mechanistic impact. So this gets back to showing if there is a targeted response or if there is a polypharmacology, and dissecting out what that activity is. But collectively, if these models are designed correctly and the phenotypic models are appropriate, one has a much more translational outcome, because you've evaluated the complexity of biological activity and phenotypic responses as a core part of your drug discovery pathway rather than as one of the steps. And so this often relates in a much more translational outcome that, if these models are correct, can reflect what one will typically see in a clinical trial setting. So ultimately, this can help improve the chance of success in clinical trials. So just a slide here to show where some of these examples could provide impact in terms of being closer and being more translational translational in terms of predicting clinical outcome. And this can really happen at a variety of steps throughout the drug discovery continuum. So target-based approaches, as we'd mentioned, typically enable a rapid way of screening molecules, whether it's compounds or otherwise. The phenotypic approaches are very much unbiased. And like I say, they do not look at a particular target, but they look more so at the biological outcome, which is, after all, what is going to happen in a clinical trial. So that may show, or it may identify aspects of targeted drug discovery, or it may identify polypharmacology and, as I mentioned, safety issues. So typically phenotypic and translational studies go hand in hand. But as I mentioned, if you start here, you're closer to the phenotypic outcome, and there are being currently put in place advances where phenotypic drug discovery can be used in much higher throughput manners and thus enable some of these screening activities. And one can always go back and then identify the mechanistic activity of how a particular agent is working once it has been identified and optimized for its phenotypic properties. So this is just an example of a well-known agent and how phenotypic approaches can show broader activity. So what we're looking at here is paclitaxel, which is a very well-known anti-cancer agent. It's typically used as a cytotoxic microtubule disruptor, and it is well known for that. So therapeutically in the cancer field, it is used for its ability to preferentially disrupt microtubule formation and dynamics, which is a fundamental part of cell division. And of course, in cancer cells, which are growing and dividing uncontrollably, it has been proved to be a very valuable therapeutic in that respect. But when one looks at a complex phenotypic model system, and this is an example using the Eurofins Biomap Diversity Plus system, we can evaluate paclitaxel here, and this is actually showing non-cytotoxic activity at the tested concentration. So these are very low concentrations here, but one can see other activities of paclitaxel. So I'm not going to go into great detail here, but you can see modulation of inflammatory properties, and here in terms of fibrotic activities. So when you actually look at paclitaxel here, what you are uncovering is that it has components of its activity that decrease inflammation-related activities and a variety of other activities in these complex translational, in many cases, multi-cell type co-culture models. So what you're identifying here then are other properties of this agent that ultimately can and do contribute to the therapeutic efficacy of paclitaxel. In fact, so much so that when you look at some of these activities in a clinical setting, you do see the manifestation of these activities. So a good example here of retrospectively how a phenotypic approach can identify essentially a polypharmacology within a known established agent that has long been thought to act solely through the microtubule complex. As I'd mentioned, one of the key things about phenotypic approaches is that you can reveal clinical toxicities. And again, better to be able to resolve this earlier in drug discovery, and especially in early lead optimization, going into lead optimization, where you have active SAR modifications being made that are optimizing those agents as you go forward towards candidate selection. In fact, even earlier, and for thinking like about something like small molecules, if you are at that junction of selecting particular lead series, knowing toxicity liabilities of one lead series over another may be very important and potentially very useful in helping delineate which lead series to follow as one goes into lead optimization. So just some examples here of the kinds of toxicities that one can not only model but identify from when one evaluates an agent in a complex phenotypic system. And again, this is piggybacking on the previous slide that looked at paclitaxel. So you can see here that there are some of the immunosuppressive activities that one sees with paclitaxel. and potential also organ toxicities here. So this identifies what we know, and again, as I said, what we see with paclitaxel going forward. So one can then model this and put this into a hypothetical drug discovery program where one is developing a new drug, and knowing some of these activities earlier, one can do something about that. Another important thing going forward with phenotypic assays is becoming a bit more standardized in the approaches that we use. So I'm not going to go into great detail on all of the different pluses and minuses here, but there are a lot of approaches that people have been evaluating avidly over the last several years and that are beginning to come to the fore. We've typically used cell lines historically for drug discovery, so there has gradually been a move away from cell lines to a little bit more focused now using primary cells. One of the key advantages of this with primary cells, and the Biomap platform that I showed previously makes extensive use of this, is that primary cells are a lot closer in their biology and their physiology to how cells would be seen or would behave in an in vivo setting, as opposed to cell lines, which have often become wired differently. They are still very good models, but they have limitations in that sense. And then now we are seeing the emergence, or we have over the last several years seen the emergence of stem cell models, where we are differentiating stem cells into desired cell types. And this is particularly important because it actually allows us to generate cellular models in what have been typically very challenging biological areas, not least of which is the neurology field. So obviously isolating human neurons for culture is not as easy as isolating primary cells from other tissue types. So the use of iPSCs here to differentiate into neuronal cell types has been very, very important in that. The other thing is the paradigm of going from 2D to 3D models. So there have been strides made ahead here in terms of using organoids. So where one is using mixed co-cultures in certain cases. Use of 3D printing, so looking at bioprinted tissues, is another one of the emerging technologies in the 3D world. And then tissue on chips, so or organ on a chip type approaches. And again, looking at these becoming more complex in not only the cellular consistency of these models and the complexity of these models, but as you can see, going from these very primitive cell line type models to something that not only has more 3D structure, but contains different cell types that are more representative of how a target cell type or a cell type that is ultimately to be modulated to treat a given disease would look in a biological and ultimately an in vivo situation. So a lot of these areas are still emerging and being developed, but they are very much on the horizon, and I think will definitely form part of the immediate future in terms of where the phenotypic drug discovery world is going. So just to summarize here, incorporating some of these technologies to address complex biology or phenotypic biology is very important. Cell line development, that's obviously a key thing. So as we just seen on the last slide, going from 2D to 3D models, then ultimately to more complex models such as organs on a chip, including models where there are now microfluidics that obviously provide that ability to change the environment of the cells as one might see in an in vivo system with blood flow and lymph flow and things like that. A key part of these models is to be able to use them in a high throughput setting. Traditionally, that's been one of the big limitations of these models. But again, advances are being made where a lot of these models now can be used in much higher throughput. And so that can obviously be automated, and that allows them to be used earlier in drug discovery. High content analysis has obviously transformed some of the aspects of phenotypic drug discovery. And we are now beginning to look at areas where high content analysis and things like cell painting are being considered, especially when coupled with things like machine learning and artificial intelligence approaches and the era of big data. where then ultimately we can model lots of different changes. And again, the whole complex of polypharmacology and complex biology in terms of multi-parametric imaging really comes into its own. We've obviously had tremendous strides forward in genomic applications, in particular with things like CRISPR, siRNA approaches, and of course, many of these now being used as therapeutic modalities in their own right. Things like transcriptomics, and so looking at protein changes, mRNA changes, tied in closely with that would be proteomic approaches, and looking at changes from a much broader perspective. And again, this whole notion of rather than looking at a single gene change or a single target, looking at multiple targets or gene changes concurrently to get a fuller picture of what's happening with the biology. I'd mentioned proteomics obviously falls into that area. And really when one thinks about all of these technologies, they are all helping to enable that evaluation of multiple readouts to really flesh out a much fuller picture of how agents are working in the complexity of a cellular biological context. So just looking here in terms of how phenotypic approaches can allow development of a drug discovery program and how that can evolve. Phenotypic assays now are no longer relegated to sort of the end point of drug discovery just before clinical trials. We can really use them at pretty much any stage in drug discovery now, including target validation or initial assays. We can use them in earlier screening. As I'd mentioned, there are now techniques that we can use various phenotypic assays in much higher throughput than historically we've been able to do. And we can start employing things like co-culture models and 3D models earlier to support drug discovery. And ultimately now, what took the place of some of these simpler phenotypic assays later in drug discovery are things like tissue on a chip, organ on a chip, and so forth. And really the only thing limiting these right now from being able to be used earlier is their throughput. But I'm pretty sure that time will show that ultimately we can configure these assays where they can be used in much higher throughput, which was typically the domain of some of these other phenotypic models. So just an example here of where this kind of pathway can be used is the approval of JAK inhibitor tofacitinib for the treatment of rheumatoid arthritis. So examples here of where phenotypic models can be used earlier. Mechanistic cellular assays here, for example, looking at JAK3 mediated STAT5 phosphorylation in an IL-2 stimulated cell line. So this is in an endogenous situation where one is looking at the effect of an agent, and obviously looking at the effect on T cell biology from a sort of hardcore phenotypic perspective, ultimately going into translational models. So a good example here where multiple phenotypic approaches have been used throughout the drug discovery process, ultimately resulting in approval of the drug, and as I mentioned much earlier, clinical adoption. So just to set this in a historical context then, in terms of phenotypic approaches, it raises the question, are we seeing a renaissance in this? When one looks at earlier drug discovery, a lot of those drugs were often discovered by phenotypic approaches because there really weren't the technologies available to look in much more targeted, focused perspectives. And this is particularly true when one looks at the anti-cancer agents. A lot of the old cytotoxic agents were identified based on phenotypic observations. Now, as we've gone through the 1990s and right the way through to the 2020s, in effect, there have been tremendous strides ahead, in particular in molecular biology approaches, but also some of the assay technologies that have allowed a much greater focus and a much more effective and an efficient way to look at things in a very targeted manner. So this has really been the era of targeted drug discovery, and we often hear the term targeted or personalized drug discovery based on specific targets. So we've gone from typically things that are first in class that are perfectly efficacious, but often have safety or side effect issues, now to going to things like best in class that are safer drugs, but nonetheless targeted. But with some of the emerging technologies that I'd mentioned, and especially the ability to look at some of these safety aspects that were limiting with some of the earlier drugs that were discovered, can we now identify and design, and especially with big data and bioinformatic approaches, looking at wider biologies, can we then potentially, and are we now on the eve of a resurgence in phenotypic approaches that really use the benefit of what was seen earlier, but with these new technologies to then resolve some of the issues that have plagued earlier drugs? So with that in mind, I'm going to hand over to my esteemed colleague, Cheryl Denker, who will give you an overview of fibrosis as a case study for how one can use phenotypic approaches in drug discovery.
Okay, thank you, Alistair. Give me a minute. I'm going to share screen. I'd like to do it that way because I want to use my pointer. So.
No problem. So I will.
And I think I will go off camera for bandwidth's sake.
As will I.
So can everyone see the PowerPoint?
Yeah, we can see it.
Okay, perfect. Get this. Okay, great. So yes, so we are going to look into this complex disease, fibrosis. It's really a disease of both inflammation and tissue remodeling. Really, in the disease state, it's really a condition of prolonged inflammation and aberrant tissue remodeling. These are processes which happen all the time. However, when there's excess inflammation, we go through this pathway, and we'll go through this figure a little bit. So there's a lot here, but as we walk through it from the left to the right, you'll see what the processes are and how they relate. So when there is, inflammation that's caused by injury, infection, even metabolic dysfunction, autoimmune conditions, or vascular disease, as you can see on the far left panel, we have a situation in the disease state. So this figure really is looking at the common aspects of fibrosis, which go across the top, and then, across the bottom, and distinct mechanisms which go across the top. So in all of those instances I mentioned, vascular disease or injury or infection, these are really sort of distinct mechanisms. It depends what the disease is or where the site of injury is. So these are considered, distinct. But regardless of what happens in this situation at the top, if we move down, then staying on the left-hand side, but moving down to the bottom, we see that there are some common reactions that happen to the biology, in particular, activation of macrophages and these other immune cells. So early on, there's an innate and adaptive immune response in the situation of fibrosis. So then moving across to the middle panel, where you see the black arrow at the bottom, into the middle section, where we have fibrosis and fibrotic remodeling that happens. And again, fibroblasts are activated to become myofibroblasts, and this is sort of context dependent as well. So depending on the site, you have a different source of fibroblasts, and this modulates the disease outcome in different tissues. Some of the common pathways, regardless, again, of the site of the injury, are listed in the bottom, and in particular, TGF-beta has been a focus for therapeutic intervention. And then again, regardless of what happens in this specific or especially shared space, as we move farther over into the far right-hand panel, we see that really what happens in fibrosis is there's a self-amplifying activation loop. So there's inflammation, defective remodeling in the extracellular matrix that's indicated there by those hash marks. And what happens then is there's a dysfunction. So that tissue can't then be flexible and have its function of filtering or support, depending again on the context. So really, fibrosis is this complex disease. It can start out as reparative in the sort of non-pathological state, and that's really beneficial. But then the disease state is initiated from this, either by repetition, either repetitive injury or repetitive exposure. If you think about smoking, there's a repetitive exposure there resulting in lung fibrosis, ongoing insult, and genetics also contributes here. And so then the whole process becomes progressive. And so we're going to look at two case studies. One is in the eye related to glaucoma, and then another is related to idiopathic pulmonary fibrosis, which Alistair mentioned. We have some instances of fibrosis on the rise after COVID, and this is a perfect example. Pulmonary fibrosis is really a consequence of severe COVID. And in particular, what they know now is that it's not just severe COVID, but additional comorbidities such as diabetes, hypertension, or cardiovascular disease. So starting here, this is open angle glaucoma, which is really a disease of fibrosis. And let's just focus on this green schematic here on the left. And what we have is the eye, a model of the eye. And you can see here, maybe you can't see, this is why I would like to have the pointer. Right here is where we have this trabecular meshwork. And so what happens in the eye, and this gray surrounding here models the cornea. And then this is the iris here. And so what happens in this space right here where the trabecular meshwork is that the fluid needs to release, and it comes out through this section, this very small area here. So the aqueous humor needs to flow out of the eye, and this keeps the pressure in the eye constant. Open angle glaucoma, as opposed to narrow or closed angle glaucoma, refers to this angle right here. So in between the iris and the cornea, whether this lens is displaced and pushes this closed, or whether the lens more so stays in the position that it is and leaves this as an open angle. So in fibrosis then, there is, and I would like to say that, 76 million people globally. Some of the latest data that I saw have glaucoma. It's one of the most common causes of blindness, and the open angle here is the most prevalent form of glaucoma. So what happens, there's an inflammation, and as we saw in the previous slide, this sort of feed forward cycle where you have fibrosis and then a stiffening of this trabecular meshwork here. So there's reduced outflow through here, and then you get a buildup of fluid and you have increased intraocular pressure. So in the case study, and I have the reference for the paper here, so everyone can go ahead and look at that. So there's a small low molecular weight dextrin sulfate that this group is looking at to change the conditions of the extracellular matrix and enable flow to again occur through the meshwork and reduce pressure. And this is important because when you have high pressure here, it pushes on the optic nerve and causes damage. So again, just reiterating, this tissue here, it's under constant mechanical stress from the fluid. This continuous remodeling of the meshwork is crucial to maintain the normal drainage and intraocular pressure. And then in fibrosis, when you have this feed-forward cycle, there is excessive deposition of extracellular matrix molecules within this trabecular meshwork, so increased resistance to the outflow here of the aqueous humor, causing the hypertension. What these investigators did, again, looking at this low molecular weight dextrin sulfate, which is known to have some antifibrotic and anti-inflammatory potential, so they knew that going in. And considering that antifibrotics alone are not successful for glaucoma, they took a phenotypic approach, as Alistair mentioned. It's very good for complex diseases, especially when you're looking at polypharmacology or multimodal drugs that can look at multiple signaling pathways. So they found, in a nutshell, that with this molecule, using not only the phenotypic approach, which I'll go into a little more detail, but they also looked at Schwann cells, downstream effects of TGF-beta signaling. They looked specifically in trabecular meshwork cells, so a more complex model, and then also moved to in vivo into a rat model of glaucoma. But this is where they started and gave them a big advantage to see. Again, these are 3 of the models that Alistair showed from the Biomap Diversity Plus. We have this model on the far left, which looks at T-cell activation of the B cells, so highly immune relevant. The middle panel here looks at arterial smooth muscle cells in the inflammatory state. And then the far panel has biology that reflects tissue remodeling. And you can see some of the activities that they uncovered using this complex phenotypic approach. So for example, inflammation-related activities, so decreases in TNF-alpha and some of these other molecules here, I'm not going to go into it, and increased tissue remodeling activities. And in some instances, these results would be indicative of antifibrotic or profibrotic indications. And you can see there's a nice dose response, concentration dependence in this assay. So again, the reference here, if you'd like to go into more detail. Another aspect where phenotypic can really help in terms of fibrosis, and really an active area now, post-COVID, is repositioning. So some of the repurposing challenges that we have is identifying potential indications and also how to prioritize the drugs. So if investigators can combine both phenotypic and target-based information, this allows them to streamline their approach and increase the chance of clinical success. Well, this is what it's all about. And then also to help prioritize drugs. So again, phenotypic with the analytics can provide a side-by-side evaluation. And I think in the interest of time, I won't go into too much detail on here, but if we start actually on the right and just look at the figures. So this then is that Diversity Plus panel that Alistair spoke of, and some of the systems that you might recognize from the glaucoma study here and here. So what these investigators did then is to compare approved IPF drugs, so that was nintedanib and pirfenidone, with a couple of their candidates, and looked for sentinel activities that would be antifibrotic activities. And then they were able to rank their drug candidates on these sentinel activities. And then from that, take their top priority candidates and move into a more specific, more detailed type of phenotypic panel, which is shown here as the Biomap Fibrosis panel, to move in to look at more specific markers for fibrosis. So basically, this allowed them to take a small library of candidates and quickly pivot to this new indication, and to do so in, using a system that shows complex disease. As Alistair mentioned earlier, the benefit of phenotypic is really this complex approach. And again, allowed them to do to rapidly prioritize for a new indication, and also to consider safety data. So from these two sort of overall overviews of these case studies, we can see that, as we understand, fibrosis is a disease of tissue remodeling and inflammation. Often a multi-pronged approach is needed. Target-based and phenotypic both have their benefit. And sort of in the field, people are considering, with fibrosis on the rise for many different reasons, do we have enough therapies existing that are adequate to address this disease, which has so much complexity? And then in the future, in particular for IPF, treatment may be a result of combination therapies. And this is something that phenotypic approaches can also handle, combination testing, to see what is the biological outcome of combinations, which is something that's more difficult to do in terms of assessing the biology in sort of target-based approaches. So I think, Alistair, I am going to pass it back to you.
Okay, thank you, Cheryl.
Sure.
So share my screen again. So everybody can see my screen here. I'm just going to go through some of the challenges that remain with phenotypic drug discovery. So we've seen where there has been value in a certain number of therapeutic areas. There's been significant success in the therapeutic areas outlined here. And we've seen this in terms of improved efficacy, better safety, and ultimately superiority of the drugs that have been approved. But the question then is where can we go, and where can there be greater success? So if we look at models here and looking at other disease indications, what we've really got to be able to get to grips with are better models of human primary cell types. As I mentioned, for neurology, that's quite challenging, obviously because of the inability to take out primary human neurons. That's a good example there, where we have to find ways of modeling the biology that are more translational, but also practical. Relevant stimuli, so understanding some of these diseases in other therapeutic areas, and again using neurology as an example, some of the fundamental underlying causes in some of the neurodegenerative diseases are not always fully known. Neuroinflammation is obviously a key part in many cases, but there are still some certain unknowns in those diseases. And obviously tied in with that is identifying clinically relevant biomarkers. It's one thing to follow a particular marker, but if it's not a clinically relevant biomarker, then it doesn't really give you the value that a clinically relevant biomarker would do. And obviously it's a little bit of a chicken and egg situation, but having a clinical drug validation is very important, because that can then be used to validate these models in a phenotypic setting. So, as I'd mentioned, areas where we are still a little bit behind in terms of phenotypic drug discovery. CNS, I give them some examples there. Aspects of liver disease, NASH obviously being one of the major areas of focus. Metabolic diseases, still in some cases diabetes, but there are being strides made in this respect, looking at metabolomics, and again tying in with that idea of big data. Acute heart failure and certain genetic diseases obviously are other areas where we need to be able to generate better models. And obviously some of the strides we've made forward can potentially be used to generate models that are better for these. And this bottom one is actually particularly challenging, where one is looking at aging, or in the example here, muscle degeneration, because there's a temporal component to the models there. So it's one thing to put in place a model that represents a particular disease state, but when you've got aging, the question then is, do you have to age the in vitro models that you're using? So again, some of the approaches that we've discussed, we talked about iPSCs of various kinds, but also ESCs, and then some of the more complex models. And again, the stimuli and understanding the disease is of paramount importance to getting these correct. And these are not easy models to set up, and for good reason, because they are complex by nature. So being able to identify that full level of complexity and to model that in vitro is key to their success. And tied in with that, of course, are the biomarkers. And obviously, because these have been challenging models to put in place, and in some ways challenging therapeutic areas, the number of drugs available are much fewer than some of the more widely studied and successful areas. So again, just coming back to looking at how phenotypic assays can impact preclinical programs, one can really use them at any stage. And I'm not going to go into great detail here, but suffice it to say that with the right models and increases in throughput, one can use it really at any stage in drug discovery. And I would go as far as to say that one should, because one can uncover some of these complex aspects of biology and of safety and toxicity signatures much earlier in drug discovery. So obviously this ensures that the candidates being taken forward into clinical trials have the very best chance that they do of clinical success, and also potentially has the possibility of maybe shortening the drug discovery pathway as well. So in summary, just to wrap up then, one of the key advantages of using phenotypic drug discovery is to really try and avoid or limit or even get away from the progression of poorly qualified leads to ultimately to candidate selection, possibly even getting into clinical trials where ultimately they are doomed to failure. So there are a lot of pipelines currently that are overloaded with leads that may not necessarily be the best that they can be. And we feel that the use of phenotypic approaches here will improve that chance of success, may allow the pipelines to become a lot more streamlined. Obviously, a key part of this, as I'd mentioned previously, is getting rid of some of those undesirable mechanisms. They may be individual mechanisms, they may be polypharmacologies, but collectively they result in things like safety and toxicity signatures. And so getting rid of those earlier can obviously improve the chances of success, but also reduce the cost associated with drug discovery, which currently is phenomenal. And then obviously advancing prioritized leads with a higher potential for disease modulation, and especially if characterized in highly translational preclinical models, greatly improves the chance of clinical success. So obviously that's a benefit for those developing the drugs, but perhaps most importantly, a benefit for those receiving them. So again, there are lots of different capabilities along the drug discovery pipeline, and I'm not really going to go into any great detail here, but you can look at some of the aspects here and think about where phenotypic models can provide particular impact depending upon your particular area of study. So really at any stage, really fully fleshing out how biology is being modulated, either in early discovery from more of a target validation perspective or initial identification of hits in hit finding or lead series selection, obviously classically in lead series, and then ultimately in preclinical evaluation. So really, if employed correctly and appropriately through all of the stages of drug discovery, there's tremendous benefit to be had. This is just a note here, some more examples of where different aspects of what we in particular at Eurofins can offer by way of different phenotypic approaches can be used and are used by a wide variety of clients, from academic institutions right the way through to small biotechs to big pharma. So people are successfully using phenotypic drug discovery at all stages in drug discovery now. We think the horizon looks tremendously bright in terms of this and improving the quality of drugs that are being put into clinical testing and ultimately approved. So I'm not really going to go through this because in the interest of time, I just want to wrap up, but suffice it to say there are lots of different models and capabilities available, not only within Eurofins, but within other companies as well. And collectively, I think as part of the pharmaceutical industry, this is really helping to transform the way in which we're doing drug discovery. So with that, I know we're about at time, but I'd like to thank you all very much for the distinct privilege of being able to present to you, and be quite happy to answer any questions that anyone may have.
So thank you, Alistair and Cheryl, very much. Very interesting presentation. Before we go into questions, I just want to say, everyone, that so we don't have much time. I'll just ask one question, but if you have any questions that are left unanswered, feel free to get in touch with us. My email is in the meetup group. So we have a question: how does the authorities look at phenotypic assays, and is it something that the regulator is expecting to see in a dossier?
Yeah, that's a great question, and that's always a key thing for getting drug approvals to go into clinical trials. We have, even in recent years, and maybe even to an extent in past years, seen phenotypic approaches be used as part of regulatory requirements showing activity in cellular models. Those models are now becoming more complex. There are, at least to my knowledge, currently not demands for specific complex co-culture models and things like that. But I think in terms of putting together an IND package, it provides more benefit and it provides more impact to those packages that makes it more likely for the regulatory agencies to take them on board and to say, yes, here is approval to take this forward into a clinical setting, especially if those models are highly translational, and especially if those models have a track record of predicting clinical outcomes. One quick example of that is, and it's a simple one, is the employment of cytokine storm assays in biologics drug discovery. So historically, that wasn't something that was included, and unfortunately, with some of the issues that were faced with the trial of one of the anti-CD28 monoclonal antibodies, it stirred interest in the potential to get cytokine storms into drug discovery pathways for biologics. And in fact, now that is something that I always say to people, while regulatory agencies like the FDA don't mandate it, they strongly recommend it, which is about as close to a mandate, I think, as regulatory agencies do get. So a good example there where there was a need for a phenotypic approach that now has been embraced and is providing value in providing safer drugs going into clinical testing.
Yeah, just to quickly add to that, these models also do help the FDA, align with guidelines for reducing the use of animal testing.
Yes.
So this is also a key component. Again, like Alistair said, maybe none of these assays are particularly required, but they do align with those guidelines here, obviously in Europe, where they started.
Okay, so again, thank you both very much. It's been a pleasure hosting you, and thank you everyone for joining us.
Thank you.
See you
Thank
next
you all.
time. Bye-bye.
Take care, folks. Bye-bye.